going to have to start again. So I'd like to welcome everyone. This is Dr. Raphael Nemenoff, and Dr. Raphael Nemenoff is at the University of Colorado. He was one of our 2018 Longevity ALK Positive awardees, and we're extremely grateful to Dr. Nemenoff for having taken the time to present at our summit. We're very apologetic that the time was so limited and his presentation was so rushed. And there was an overwhelming demand from hundreds of our members to hear more details on the presentations from the panelists. So Dr. Nemenoff very, very gratefully has agreed to spend about an hour this morning discussing immunotherapy and ALK and his project. And he'll have time to go into scientific detail, but at the same time, explain it in layman's terms. So Dr. Nemenoff is going to do a bit of a self-introduction and then move into his presentation. And he's given me permission to interrupt him at different times. And I'm just going to turn on the share screen so that Dr. Nemenoff can make his presentation, and I'll hand you over to Dr. Nemenoff. Thank you very much.
Okay, thank you again. I wanted to, Colin, thanks a lot for this opportunity. I actually really enjoyed the ALK Positive Summit, and I really, again, want to reiterate our appreciation for my lab and all the people I work with for the support we've gotten from Longevity and from the ALK Positive group. And I think we've known Ross Camidge for a long time, and so I know that he's a bit of an icon in this group, and it's been a pleasure. So just for myself, I've been at the University of Colorado for longer than I care to tell you, but it's more than 20 years. I'm a basic scientist. My PhD was actually in chemistry and have sort of drifted over the years into more and more translational studies regarding lung cancer. And this was really facilitated by becoming part of the lung cancer SPORE, which is a large multi-investigator translational grant from the National Cancer Institute, which was headed by Paul Bunn for many, many years, and has now been taken over by Bob Doble. And I've been a project leader in that for, again, longer than I care to admit. And this is really, I think for me, it's been really a journey of, for what you do in the lab and sort of being able to actually see how this impacts patients. And for me, the SPORE, which is, a coming together of people who do basic science, people who do translational studies, people who run clinical trials, I think the intellectual interactions really make it for a really exciting environment. And I've learned, a tremendous amount in dealing, in sort of relating what we do in the lab to actually trying to move this ahead into clinical practice. And, the real accomplishment here would be to try to develop new therapeutic approaches. The project I'm going to tell you a little bit about kind of came around in a serendipitous way. So complement, which I'm going to tell you a little more about, is part of the body's innate response to infection and cancer. And a colleague in my department, in the Department of
Could you explain what you mean by innate response, please?
Yeah, so in the setting of infection, your body responds in two ways. So there's a quick response if you have a viral infection, and we'll apply this to cancer in a minute, but there's a level of response that immediately recognizes something bad is happening and mobilizes a number of cell types, a number of processes to try to deal with that. So if you have a bacterial infection or you have a viral infection, such as COVID, you basically have a quick response which happens very, very quickly within hours. And it's kind of, you can think of it as sort of the first wave of soldiers out there. Now, this is a response which is not as specific. Your body also has an immune response. And as you may know, the immune response, which is mediated by lymphocytes called T cells, actually is very specific. So T cells in your body actually recognize specific bad things about foreign substances, viruses, tumors, bacterial infections, and mobilizes a very, very specific response. So, for example, all the vaccines that are being made, that are trying to be developed for COVID, are designed to mobilize this T cell response. And so the T cells will recognize something very specific let's say, virally infected cells and kill them. But your body also has a quicker, less specific response, and that's the innate response. It's called the innate immune response. It happens more quickly, and it precedes this more sophisticated but slower activated adaptive immune response. And the two communicate. So the complement pathway, which I'm going to talk about in a few minutes, really represents part of that early response. So if you have something happen, like you have an infection, complement pathway is one of the things, the way your body responds to try to get rid of that or try to neutralize it. It's one of many things. There are inflammation and recruitment of inflammatory cells. So there's this very complicated attack on these foreign substances. And the complement pathway is one of the pathways. It's been around for a very, very long time. And evolutionarily, it's probably one of the oldest components of the way our body deals with infections. So I had a colleague named Josh Thurman, who I've collaborated with now for the last 5 or 6 years, who actually has studied this pathway, but in nothing to do with cancer. And he's studying it in terms of kidney diseases and chronic inflammation. And we just happened to chat. And I think this is one of those things that happen, it's, not that often in your career where you actually have a conversation with someone which kind of inspires you to move in a new direction. And after we sort of chatted over coffee for a bunch of times, we thought about, well, should we look at this perhaps in our lung cancer story? So we've been studying lung cancer at in preclinical models for quite a long time. But about 4 or 5 years ago, we decided to join forces and begin looking at the role of complement. And clearly this has been looked at. We're not the first people to look at this. And this project has evolved over the last 3 or 4 years, and support from longevity was incredibly critical, I think, a watershed in terms of actually getting resources to do this project, and has been instrumental in us getting an NIH R01 to do this, not just in ALK positive lung cancer, but extending it to other oncogenic drivers in lung cancer. And this has now been integrated into part of our SPORE project. So I think we're working together with Ross Camidge and other people to try to actually move this forward into actual clinical trials. Because
what a NHI 101 award or grant is, please.
I beg your pardon?
Could you explain? You mentioned.
The sort of a major investigator-initiated grant from the NIH is designated as an R01 grant. This is a grant which actually is to both myself and Josh Thurman. It's a 5-year grant for over $350,000 a year. And so it allows us to really expand this project. And and that grant is actually doing very different things than what we've been able to do with the support from longevity, but they're complementary. And we're now trying to move this even into more translational studies. And a lot of, what we've been doing in the last 6 months is actually having conversations with pharma and having conversations with some startup companies that are actually developing inhibitors in this area. And I think there's some optimism, more than I had 6 months ago, that in fact this will go into a clinical trial in lung cancer, not just lung cancer, but lung cancer for sure. And in fact, to my pleasure, we have now initiated at our institution a phase 1 for a novel complement inhibitor. And I'll go through the pathway in a minute. But I think there's really optimism that these drugs at least will go into clinical trials in a variety of cancers, in a variety of scenarios, and I think offer new opportunities, particularly for ALK positive lung cancer, because at the end of the day, what our interest in the lab is, and what the interest of this project is to really target the immune system to attack cancer cells. And so immunotherapy, which has been an idea that's been around for 100 years, sort of says that the immune system actually can recognize cancers and can theoretically eliminate them. And for a long time, this was a nice scientific idea, but as I'm sure most of you know, the advent of immune checkpoint inhibitors, which has been developed in the last 10 years, has really revolutionized the treatment of many cancers, including lung cancer, but certainly not exclusively, melanoma, kidney cancer, etc., and has really provided a new day for for the treatment of cancer, because one of the big deals about this type of therapy is not only does it work, but if it is successful, it works for a long time. And there are certainly patients, and I have a personal friend of mine who has a Merkel's cancer, who has now been successfully treated for 6 years with these therapies and is cancer-free. And I think this is really exciting. However, these things don't work.
Can you clarify, would you say that that patient is cancer-free, or would you say that...
At the moment, he's cancer-free. So this is a close friend of mine, and he goes in every few months, and they check to see, and so far he's cancer-free, and they've actually stopped the therapy. And this is, I think...
How long has the therapy been stopped for?
It's been now stopped for almost 2 years.
That's amazing. So that's something that is, to our members, I think, such an incredible concept, because, as you know, at the moment, once we're on treatment, we're on it. And so
I think for ALK, I think if one could harness, and maybe we should spend 2 seconds talking about this. So in ALK positive lung cancer, the immunotherapies that have been approved to date for the FDA really don't work very well. However, that does not tell you that immunotherapy is not going to work. It tells you those therapies don't work.
Right.
I think the ability to engage the immune system and to actually treat and potentially cure, and that's a word that I think, Ross has used that word occasionally, and it always, I think the first time he actually used it in a talk, people kind of looked at him, because you don't actually think that we actually have the ability to cure cancer patients. But I think there are, and it's certainly not everybody, but immunotherapy has had the ability to cure a number of cancer patients, both in lung and in other malignancies. And what I mean by cure is basically you're cancer-free, they stop the therapy, you continue to be cancer-free, and the data's, this is a new therapy, but in the case of melanoma, they followed patients 4, 5, 6 years after stopping therapy, and they're still cancer-free. So by all practical purposes, that's kind of a cure. Who knows where the future lies, but I think this has really excited investigators and clinicians to really try to understand how the immune system works. And it's not going to be, we're really in the early days of understanding the interactions between the immune system and cancer. And the fact that immune checkpoint inhibitors that have been approved, like pembrolizumab and nivolumab, that they don't work doesn't mean that the immune system is not being engaged. It just means that particular pathway is not effective. So the goal of our project really is to try to understand how to engage the immune system in ALK positive lung cancer.
Would you say that a reasonable analogy might be like saying that a EGFR TKI is not going to be effective for an ALK patient?
Absolutely correct. And again, I think, I think our understanding of how the immune system interacts with, specifically with ALK positive and other oncogenic drivers, is still in its infancy. We know a little bit about this, but this is a new area of investigation. The advent of these successfully FDA-approved drugs has really sort of, fired up, I think, people to look at this. And I think there are many, many approaches to try to engage the immune system to eradicate and combat tumors. The complement system may represent one of those pathways, and we're focusing on that. We are certainly not the only show in town, and there are certainly lots of other people looking at this, and there are combination trials, as you may know. There are literally 3,000 clinical trials ongoing in cancer using a combination of a checkpoint inhibitor plus something else. I think some of those have shown promise, some of those have not, and I think it's a very, very frenetic and exciting time. I think it's a little bit like, when there's chaos, there's obviously things that are happening. This is not a quiet field. This is a field where there are tons of new drugs, tons of new opportunities, and I think that's, in Ross's talk, he talked about hope for the future, and I think there's tremendous hope, because I think there are new approaches that have not been, looked at ever before, and are having success in some cancers, not others. And I think in the next 5 to 10 years, we will develop a whole bunch of new therapies, probably combinations, for ALK positive lung cancer. So that's kind of where my feeling is. And what we're trying to do in our group, I think we're trying to look at this at being a basic scientist, we're really trying to understand the mechanisms and trying to use those mechanisms to develop rational approaches, either to give one drug or combinations of drugs, but in our preclinical models to reproduce what's going on in the patients, and then to look at patient samples and to try to correlate that and see if that tracks and if we can then move that into clinical trials. So obviously, I'm not an MD, and one of the challenges for this project, which was discussed at the longevity meeting last year, is can we actually do this? Can we actually get pharma interested in these drugs? And that's been an up and down road, because there are complement inhibitors, which I'm going to talk about in a few minutes. They are approved for other diseases. We've spent quite a lot of time trying to talk, Ross and other people in our institution, to talk to pharma, and to, some of the companies, to be honest, are not that interested. They've got a drug that works, they are making money, and they're not that interested in moving into new areas. However, we've also got some startup companies that are developing new drugs, and there are now a greater accelerating interest, I think, in complement inhibitors actually going in clinical trials. And one of them just opened in our institution. It's not lung cancer specific, and it's just a phase 1 with a new drug, but I think there's really hope that that will move forward rapidly. So that's kind of where we're at.
Could you just give, for the benefit of some of our newer members, give a really brief distinction between a basic researcher and basic scientist and a translational researcher?
Sure. So, I mean, my background I actually did my degree in chemistry, believe it or not, and many, many years ago. A basic researcher really is the questions they ask are really trying to understand mechanisms and using model systems to try to develop an understanding of the basic biology. The translational component then becomes, can you take that knowledge and extend that into a study that will eventually impact patients? And so, I mean, an example of that can be actually found if you look at the immunotherapy checkpoint inhibitors. Studies done by Jim Allison's lab that eventually got a Nobel Prize were done 15, 20 years ago. They were interested in how the immune system was regulated, and they found a pathway without any thinking about patients or without any thinking about clinical applications. They found a pathway that could reactivate the immune system in several models of cancer. And they were interested in how that worked, and they were able to show that it involved this interaction between a cancer cell binding to a lymphocyte and turning it off, and they were able to reactivate that. And that was all basic studies, basic science, published in journals read mainly by biologists. And then the great advance was then to try to say, okay, this actually seems to work in a preclinical model. Can we test it? And that starts to become translational. We're now developing therapeutic agents, which will initially work, at least, let's say, in mice. So can we develop something that will either cure or contain cancer in mouse models? And that's the sort of stage 2. And then moving on to that, you then begin to talk to clinicians and try to develop a clinical trial. So there's a pathway where you start with a basic science observation. You then become translational in the sense that you're now thinking, well, maybe this actually will work in people, but before I needed to work in people, I needed to work in some kind of experimental animal model. If that is successful, you then go forward with that particular, you have to obviously commercialize this, but then you can take that particular agent and move forward into testing it in people. The first thing that you do is a phase 1 trial, which is saying, I have a new drug, I want to know if it's safe. Am I actually causing harm? And so that, there's usually a trial with a limited number of patients to show that this actually doesn't cause harm, and you get some initial inclination that it actually has some benefit. And then you go to the second stage where you're now assessing its benefit more broadly to say, is this really worth doing a large clinical trial and enrolling potentially hundreds or even thousands of patients and actually see that it works, in order to get approval from the FDA for a drug to go into clinical practice. And so in order to go through that, you first need the preclinical data generated by the basic scientists, and then you engage the clinicians to make sure the drug is safe and that it shows some promise. And then in a large clinical trial, you have to prove using, all the rigorous statistical analysis that it actually causes benefit and it doesn't cause harm. And this is actually analogous to what's going on right now with the COVID clinical trials and vaccines. I mean,
Exactly.
unlike the Russians, we are actually doing this in a more logical way. So the first vaccines have gone through a phase 1 and they're safe, and now they've gone through a phase 2, and now they're phase 3 studies to see, do these actually work? But
Right.
before they did that, they actually tested them in a variety of models, and there are a number of, they've used, for example, primate models to see, does this work at least in a monkey? So if you give a monkey COVID, does this vaccine protect? And the same thing, does a complement inhibitor prevent cancer in an ALK positive model of lung cancer? And that we have that data, and we're not the only ones that have that data, but I think there's now data from a number of labs which is sort of now finally aroused the pharmaceutical industry to try to develop these drugs and actually take them through the trials that are needed to have them actually work for patients.
Right. So tell us a bit about complement inhibitors and what your hypothesis was.
Well, maybe I should show my slides, and I can sort of, I'm not going to dwell on this, but I can go through this relatively quickly. Let me share my screen. Does that work?
Sure does.
Okay, so yeah, I'm not gonna dwell on this. I'm gonna try to. So again, we've talked about this before. So overall goals in the setting of ALK positive lung cancer, they're obviously first, second, third generations, and we know a lot about these targeted therapies that basically block ALK kinase and they show efficacy. There's obviously heterogeneity of response, but one of the problems, as Colin mentioned, is you need to keep taking them, and they eventually develop resistance. So immunotherapy is a very appealing idea because it, in the setting that it works, and it looking at all cancers and all patients with just checkpoint inhibitors, which again is one immunotherapy, the response rate is somewhere in the order of 20%. So most patients don't respond to this. In particular, ALK positive lung cancers, the response rates to immune checkpoint inhibitors is low. So these agents as monotherapy are unlikely to be effective in terms of helping ALK positive lung cancer patients. However, that doesn't tell you that the immune system can't be harnessed. And I think our approach, and again, others are doing this as well, is to develop new therapeutic approaches that target the immune system in ALK positive lung cancer. So the complement system is an extremely complicated system, and I know for the, for patients, this seems like this is one of those figures that you immediately zone out on. And I can tell you, having presented this at scientific meetings, that people who are not in the complement field, also their eyes glaze over and they look at you like there's something wrong with you. So. And that is one of the realities here. So I am not going to go through all this other than to show you this figure is a very complex figure. But what happens if your body has, let's say, a cancer or it also applies infection, there is this, even the name is bad, and it's an old name which is meant to complement other pathways whereby your body protects itself against infections. But it involves this very complex pathway, which involves a lot of different proteins that are deposited on cells that are then undergo these reactions where they're cut into pieces, and the pieces do something. And I'm just going to give you one little, snapshot of this, but this is a very complex system. But one of these things is that there's a protein called C3, and the name for it just means complement protein 3. And in the setting of an infection or in the setting of ALK positive lung cancer, this protein is actually stuck onto the cancer cells. And that already tells the body that there's something foreign about these cells. So cancer cells which have C3 on their surface then undergo this chopping of that protein, so pieces of it fly off. one part of it becomes C3A, the other one becomes C3B, and forget the nomenclature. But the bottom line is that when this is on a cancer cell, you generate these molecules which actually engage other cells to come into the cancer. So it engages inflammatory cells such as macrophages and neutrophils to come in. And it's part of the way that in a normal infection, your body would get rid of, let's say, a bacterial or viral infection. But in the setting of cancer and in the setting of ALK positive cancer, this results in a chronic inflammation. And that actually acts to promote cancer progression. So there's a lot, I'm not going to go through more of a compliment, but I need to show this because I'm going to show you a slide a little bit later about where the inhibitors, where the drugs that show promise as therapeutic agents in ALK positive cancer may be developed. So our project, which was originally defined for longevity, was actually had two specific aims. And again, we're doing basic science. We're interested in understanding how this works. So the first aim of our project was to define the mechanisms whereby complement activation mediates progression of ALK positive lung cancer. So what I mean by that is we're interested in understanding when there's an activation of this pathway in an ALK positive lung cancer, what has happened? How does it work? What is going on? And I'll show you a little bit of data in a second. And the second part was actually really getting into what I think will be some hopeful progress for ALK positive lung cancer, and actually to test inhibitors, and particularly new inhibitors. And so that's kind of the two things that we've been working on in this project. So this is data, again, that we published a number of years ago, but just tells you that this works. So I'm not going to go through the details of this, but this is basically a model of ALK positive lung cancer which we're able to use in mice. So we are able to implant ALK positive lung cancer cells into the lung. They form a tumor. They will eventually metastasize. They have all of the features of human ALK positive lung cancer. We also know, consistent with what you see in patients, that immune checkpoint inhibitors don't work in this. So we have models where they work for ALK positive, for our mouse models of ALK positive lung cancer, these agents don't work. However, complement inhibitors, so these are two different models. These are just two different ALK positive cell lines. And I'm just going to take you, so this is just where we implant the mice. We let the tumors grow for a period of time, and then treat them with these drugs. And we've done either a genetic model, which I'm not going to talk about, but we've used these two complement inhibitors. One blocks that molecule I showed you before, C3A. So this binds, that one blocks this, and one blocks, one blocks this. So there's a C3A blocker and a C5A blocker. And you can see that in both of these, the tumors are inhibited by greater than 90%.
Wow.
So this, so this tells us in this model, and again, how applicable this is to humans we're a long way, but at least in this model, these drugs are very effective. And they're effective in at least a couple of models of ALK positive lung cancer.
Now, are these drugs that are already FDA approved, or are these?
These are not. I'm going to talk a little more about this. This is a drug that that blocks C5a receptor. The drugs that are now in clinical trials, so there are these are what are called toolbox compounds. So they're not the actual drug, but they're hitting the same target. So the clinical trial that is open at our institution is working with something which will act exactly like this.
Okay.
There are the only FDA approved complement inhibitor actually also acts through this pathway. So there's a compound called eculizumab, which I'm going to mention in a minute, which is actually FDA approved for a variety of kidney diseases, and that acts on the same place. So the second compound, this one here is a small molecule which you can buy. There are currently no FDA approved drugs that work like this. And there are some being developed, but there's nothing that is FDA approved yet. This pathway, there are FDA approved drugs.
Okay. So for the drug that you mentioned that is FDA approved being used for kidney, Is the safety profile on that seem pretty good?
Safety profile is terrific. I can actually skip to that and come back to what we're doing, but let me just skip to that because, okay, so here, this is kind of, I think, the nuts and bolts. We don't even have to go there. So eculizumab it's a monoclonal antibody which blocks this. And it's approved for these kidney diseases, which I know very little about, and I'm not gonna take a lot of questions because I'm gonna look really stupid. But these paroxysmal nocturnal hemoglobinuria and an atypical hemolytic uremic syndrome are kidney diseases, which are fairly serious diseases. These drugs, this antibody, which is FDA approved, it works in these diseases. It is very safe because the patients take it for years. So the side effects of this are very minimal. I can also tell you from a economics point of view that the company that has developed this, these are designated as orphan diseases, and the therapy for this drug costs about $400,000 a year because there's one company that makes it and they've got a lock on it. And I think one of the reasons that we've had difficulty, and the field has had difficulty in sort of getting these companies to have an interest, is they've got a nice, as the word is, a nice gig. They're making a lot of money with a drug that is safe, and they're treating the disease, and they're kind of reluctant to move into areas, for example, where they might not have as much success. So that's where the establishment is. There are now a number of pharmaceutical companies. I know Regeneron has an antibody, and we've had some conversations with them. And there's this company, I'm going to show you this clinical trial right here, is from a company called IMAB Biopharma. And this is a synopsis of a clinical trial that's opened at our place. And this is a monoclonal antibody, a monoclonal antibody against C5, as I told you. And this is developed by a company which is trying to move this into cancer. So I think what has happened in this field is that the accepted drugs that the FDA approved drugs are kind of, those companies are not really that interested in moving into new areas of disease. New companies are coming along. This is one of them, but there's certainly others.
Okay.
Okay, so very quickly, I don't want to belabor all this, but so just to show you just the kind of stuff we do. And again, I'm not going to go through all this, but as a basic scientist, one of the questions we're asking, how do these things work? What do they actually do? And one of the things we believe is that when you can inhibit complement, you change the ability of all these other cells to move in and out of the tumors. And one of the things that we're seeing increased numbers of lymphocytes, T cells, coming into the tumors when you treat with complement inhibitors. And that is a very encouraging sign because in many, many types of cancers, more T cells are associated either with a better prognosis up front and certainly better responses to immunotherapy. So we actually, without going out and saying it, we think there's some reason to believe that these drugs could be used in ALK positive lung cancer patients to sensitize them to other kinds of immunotherapy. And that's, I think, the way we're thinking about this. It is unlikely that these drugs are going to be a single agent that is going to cure everybody. Our view of this is that they will engage the immune system and will allow new immunotherapies to be more efficient in patients with ALK positive lung cancer.
In the mouse models, did you ever do a combination of the C3A and
we've done a C3A and an immunotherapy, and it looks promising. I would say I think that we've done a little bit of that, and there's another group that has done a study in Europe in a similar approach, not with ALK positive lung cancer. We've done one experiment, and it looks encouraging, but it's not going to be a magic bullet.
Right. And the question I was going to ask was, did you, in the mouse models, did you look at doing a combination of both the C3A and the C5A?
Yeah. You don't really get a lot. I mean, they both, I would say, with each one alone, you get maybe an 80 to 90% inhibition of tumor growth. Maybe the two together, it's a little bit better, but it's not, again, you haven't eradicated the tumors.
Got it. Okay.
And I think one of the questions, and that's a great question, Colin, because, so there's a C3A and there's C5A. Do they both work? If you look in the scientific literature, most of the clinical translational studies in cancer have been on C5 because there are approved drugs for that. C3A has been less studied and the drugs are less developed. There's an investigator we've talked to a bunch in Australia named Trent Woodruff, who's actually developing new C3A receptor inhibitors. The ones that are out there are not that specific, and I think that's an area where there's, needs to be better sort of preclinical studies and better pharmacology to develop those.
Okay.
Anyway, so this is just, and again, this is an approach where we can just look at all of the cells in these tumors. So we can take out these tumors and we can ask what populations of immune cells, of cancer cells, of inflammatory cells change. And I'm, again, not gonna, this is not basically gonna, I don't want to belabor this, but I just want to show you that. So if you just look at this kind of map and you can just ask, so this is kind of a technique where each population is defined by a particular cluster in this two-dimensional map. So if you see a cluster get darker or lighter, that means it's changing. So this is just a tumor that is not treated with anything. This is a tumor treated for a short period of time with a C5a receptor and a short period of time with a C3a receptor inhibitor. And the question we're asking here is, does this affect changes in the immune cell populations? And again, this is a complex figure, but the bottom line is that if you look at some of these populations, a number of cells change. And in particular, we see an increase in these green populations and this orange population, maybe a little hard to see, but these are cells that we'd like to see coming into the tumor. Lymphocytes and some populations of certain innate populations such as neutrophils. So this is ongoing studies. We're trying to define which of these are important. How does this matter? We do know that the effects of the complement inhibitors involve the activation of lymphocytes, because we've done and published preclinical studies where if you get rid of lymphocytes in a preclinical mouse model, then these drugs don't work. So they're not just working by inhibiting the cancer cells the way that alectinib is working. They are definitely working by engaging the immune system. And that's what, that's why we're excited about using this in combination with immunotherapy.
Right. Mm-hmm.
Okay, so this just, again, just to show you that we are card-carrying translational scientists, we have looked at this in human tumors. And so we've stained a whole bunch of human tumors. Unfortunately, we don't have as many ALK positive as we would like, and it's been a continuous challenge, and Ross can speak to this, to actually get tissues from ALK positive tumors prior to treatment, because most of that tissue is used for diagnosis, and we've been, not been able to get a lot of it, but we've been able to get ALK positive lung cancer and been able to ask, does this complement activation happen? Or is this some weird thing that only happens in mice? But we are seeing it in human patients. So we've so far looked at 7 different ALK positive patients just at baseline. And this kind of arbitrary cutoff, which I think is reasonable, we see elevated complement in at least 3 or 4 of these. So this is an event that happens in a significant number of ALK positive lung cancer patients at baseline. Obviously, as we go forward, the other question that is really, which we've spent some time on, is, this is not going to replace alectinib or brigatinib. And so what about using this in combination? And we've done a little bit of work showing that these drugs may work in combination, although those are complicated studies. I'm not going to talk about them today. But clearly, this is something that is not limited to happening in some bizarre mouse model that basic scientists study, but is actually happens in real patients. So I really want to emphasize the utility of this pathway in real human ALK positive lung cancer. So this is sort of where we are in this project. Just to summarize, we've shown that complement activation is important in the progression and metastasis of ALK positive lung cancer in a mouse model. We've shown the inhibitors block this effect, and that this is mediated by T lymphocytes and is reflected in changes in the microenvironment. And again, that the activation is something that we also observe relatively frequently in human samples. So this is, again, this is just sort of the strategy, I think, where we're trying to look for drugs. So this drug, which targets this pathway, is FDA approved, and there are other companies making analogs of this. This is another, it's not they use monoclonal antibodies. This is another inhibitor of this pathway called PMX53, which is also being used by a lot of people. But I think one thing I just really want to emphasize is, I think one in that we have is we've been working with a startup company in Boston that is trying to develop a targeted protein. So this is, instead of using a drug that just blocks complement and blocks it everywhere in your body, can we somehow target this to a cancer? So could you give this to an ALK positive patient, and you would target this compound just to where the cancer is, just where complement is activated, and it would inhibit it locally. And that, I think, would definitely eliminate any potential side effects. And this has been developed. Initial studies were developed were published by Steve Tomlinson, and this was, this has been around for a little while, but this company is actually humanizing this agent. We're going to use the mouse model. If we would have already done these studies if it hadn't been for the COVID shutdown, but we're going to test it on our model because I think this is a very appealing drug which could be given to target complement just at the site of the cancer. So I think, overall, we're trying to look at multiple ways to block this and to see what is available from pharma and from other places and to try to move this ahead. And again, this is a clinical trial that is now happening, so there's been some success in getting this forward. The other nice thing about complement, I think, is that, one of the things is you want to have a biomarker for patient selection. So, as ALK positive lung cancer patients know better than anybody, one size does not fit all. And in terms of the efficacy, diagnosing the ALK mutation has been critical in, deciding what therapy you're on. For complement, we have actually an assay which allows us to measure how much is complement activated in blood from patients. And we validated this in mice, just showing that, that basically we can see an increase in complement in the blood from mice that have a tumor compared to mice that don't have a tumor. This is one very early experiment, but there is actually a clinical laboratory that is performing this in patients with kidney disease to designate them for complement inhibitors. And in this clinical trial, they're going to use that assay. So I think one of the nice features about the clinical trial at our institution is we're going to basically start. Obviously, this is a safety drug, but we're going to test this drug. But we're also going to see if we can use that assay in peripheral blood from patients, which is easy to obtain and non-invasive, to see, number one, does the efficacy of this drug correlate with complement activation in the patients? And if we treat with this drug, can we detect that in the peripheral blood? So this will be a terrific way of first selecting patients and then monitoring how well they respond in a non-invasive way.
When is this trial starting, and is it only at University of Colorado?
So this has gone forward. The Antonio Jimenez is the PI at our center for this, and they're enrolling patients as we go. So I think it's already opening.
Okay,
and again, this is a phase 1. It's a small study. It's really it's supported by this company, IMAB Biopharma, and it's really designated to look at safety. But they are going to look, and I don't have the second page of the synopsis, but it actually also tells you that they will look. They're going to look at mechanisms, and they're going to look at activation in peripheral blood. So I think we're going to try to get some samples for this. Obviously, this needs to be expanded, and it's certainly not the only show in town because we're continuing to have conversations with other companies as well. Right.
And then, so is the study like that, if there's a ALK positive patient that is not responding to any of the ALK TKIs anymore, would they be eligible for this?
Yeah, they would be.
Okay.
So, and again, I mean, and Ross is, obviously, this is not actually coming from Ross. So this actually came through discussions, not driven by the lung cancer group here, driven by more in medical oncology. They really sort of have a lot of experience with phase 1 trials. And so, so we're kind of, hitching onto this, but our hope would be to get other companies involved with a more specific trial, really focusing on ALK positive patients.
Great, that's exciting. So
yeah, I mean, that's basically my presentation.
More questions later.
Yeah, I mean, I think that's kind of where we're at. I think complement inhibitors represent a potential novel therapeutic for ALK positive lung cancer. Clearly, they would be used in conjunction or upon progression of ALK tyrosine kinase inhibitors. But I think the advantage of these things is they do target the tumor microenvironment, in particular T cells. And I think this has moved ahead even in the time of COVID. I think this is generating steam that there are more trials, more agents being developed by multiple companies against multiple cancers. Certainly not only ALK positive lung cancer, but I think one big, issue for ALK positive lung cancer is their resistance to conventional checkpoint inhibitors.
Mm-hmm.
So that's kind of where I'm at.
Great. Well, that was a wonderful presentation. So with the studies that you've been doing, we kind of expected it was going to be a 2-year project. We were not anticipating the interesting year that we're having in 2020 with COVID and everything. How much did that stall your research, and where's your research now, and what do you have left still to do on that project?
Yes, so we basically were shut down at the beginning of March, and we've now opened up again, in a limited way. In Colorado, things seem to be kind of under control, so we are currently at 50%. We're allowed to have 50% occupancy, so that has definitely slowed us down. I think we had some experiments ongoing. I think the major thing that has slowed us down on is actually obtaining sufficient amounts of these inhibitors to do the trials with the targeted inhibitor, and to analyze as many samples as we can. So we have some mechanistic studies ongoing. We have some genetic models, and again, this is really focused on a basic understanding.
Mm-hmm.
But it has certainly, I would say, impacted us pretty severely for probably 3 months. And I would say we're up to speed. We're not up to full speed. I would say we're probably at maybe 60% of where we were before.
Okay.
We're limited in the sense that, we are, because of occupancy, we can have, we have 2 people, let's say, in the same area in the lab, or only allowed to have 1 person.
Right.
Some of these experiments, particularly in mice, involve, getting enough experimental animals, monitoring the tumors, having all that set up. We are getting up. It certainly, I would say, has slowed us down significantly, and we're certainly not unique in that.
Right. So, what do you have left to do on your project? And given what you started off with as a hypothesis when you started the project,
Yes.
you feel like the project is proving the hypothesis? Do you overall think it's been successful?
I think it's certainly been successful. I think we've opened up. Again, I think the compliment, that slide, that horrible slide that glazes everybody's eyes over, also has the advantages that there are some other targets with it. So we've looked at some targets, and I think the major goal from the longevity project is to complete our study defining some of the changes in preclinical studies, what cells are changing and which are important. And the second part of it is to get sufficient amounts of this targeted inhibitor. We've used the other ones we have, but to actually test that in our model to see that it works.
Okay.
And then as an add-on, and this has really been, I think this came up at the longevity meeting, the challenge for us has been to get, get more samples to validate findings in patients, and that continues to be a challenge. And I think that's a challenge that many investigators, have. I think, there's so many. What we would like to have, and we have a few of these, and we have protocols open, is to be able to, assay tissues from ALK positive lung cancer patients and say, okay, I can tell you that if you have complement activation, your prognosis on, alectinib or whatever is going to be better than if you didn't have complement or something like that. And then secondly is we have a protocol open at our institution where we actually collect a second sample, and we can look at, so the way this is done, and Bob Doble and Ross have been involved in this, is so if you have a patient, you take a biopsy, you then initiate treatment. At some short period of time after that, you collect the second biopsy and you ask, how did things change in response to therapy? And can you use that to predict resistance to alectinib or brigatinib, duration of response, depth of response? We want to look at those tissues and say, how did complement change in all this. I mean, if we would find evidence, for example, that complement was activated or changed in the setting of ALK treatment, that would be really important for us to be able to design the correct rational way to deliver complement inhibitors in combination with the agents that are already out there. I mean, I don't think, as I said, this is not going to be something that's going to replace the currently approved drugs. I think it's going to work in patients who develop resistance, but, you know, are we going to give this up front? Are we going to give this at the time that they have residual disease? These are all really complicated questions which we'd like to work on. They're clearly beyond the scope of the longevity grant. But we're going on. We have other funding to continue the complement project. So the NIH grant that we have is not focused on ALK positive lung cancer, but clearly the approaches we're using and the drugs we're using are going to spill over into looking at the ALK positive. So, you know, we're ongoing. I'm very fortunate to have a postdoc who's now become an instructor who's really excited about the complement project and now driving this. And I think she's going to be, or has put in a career development award to longevity, career development award to the VA, also for additional support. So this is something we're in for the long haul.
Right. So with the project that we've funded, I think initially we, started, we thought it would probably be completed around about July or so. And obviously, I think there's been a couple of delays even before COVID came along, and then certainly COVID. Would you think by the end of the year you would have the project finished?
Yeah, I would. I mean, our hope is that we will, so if we can get in the next, few weeks, get sufficient amounts of this drug. I mean, the experiments could be performed within a month or two, and the analysis and the tissues. I would say, I mean, our aim is certainly to finish this off by the late fall.
Okay, and then from them, typically, how long would it be before you get a paper published on your results?
Well, I mean, I would assume that we would have a paper submitted by, let's say, November, December, and hopefully accepted by the end.
That's great. And then, as you said, it sounds like the hypothesis that you had when you started the project have, it looks like you'll be able to get the results that you were trying to achieve, which is never a given in research, obviously. So that in itself is fabulous that we selected a project and it has gotten to proving the hypothesis that you expected. So that's outstanding. What would be the next steps to move it forwards towards the clinic and, after this project's finished?
Well, I think there's a development of clinical trials, not just, around the world using, I think there's certainly interest in complement inhibitors in a variety of cancers, including lung cancer, in a variety of settings. I think the focus for us, and I think we're fortunate in that sense to be at the University of Colorado, is, I think the strength of the program here has really been focusing on targeted therapies in oncogenic drivers such as EGFR and ALK and ROS, et cetera. And so what we would like to do is to actually develop a clinical trial that is focused on those patients. And I think a lot of the trials that are ongoing are going to be looking across all malignancies, are going to be, not focused on specific oncogenic drivers. Right. So I think the strength, and that's where Ross and Bob Doble really come into play, is to be successfully able to recruit patients for that. So,
what would be the steps between the project that you're doing being finished and getting to, a clinical trial with?
So there's a couple things. So the targeted drug, that we're working with the company in Boston, we have little control of how quickly they will actually humanize that, and they're going to do some clinical trials in other diseases before they go into cancer. So we're kind of, keeping abreast of that. But I think the most rapid approach for us would be to have an agreement with a company. I don't know if it's going to be the one I showed you, but Regeneron or somebody, and we had conversations with Regeneron a while back, and with COVID and everything else, I think it slipped a little between the cracks, but I think those need to be reinvigorated. And to take that and do a phase 1 in ALK positive lung cancer patients for safety. It's been proven to be safe in other diseases, so I expect that will be positive, have some indication of efficacy, and then move on to a phase 2. And I think in that case, I mean, so my role in that will be more looking at biomarkers. I think optimizing, defining biomarkers in biopsies from these patients, potentially defining biomarkers in peripheral blood. There are other approaches. So I think as a basic scientist, that would be our aim, and people like Ross would run this trial and be the drivers for the clinical efficacy.
You mentioned earlier that one of the challenges that you've been facing is getting enough tissue from ALK positive patients. Do you have any suggestions on how our group could help you getting enough tissue?
Yeah, I mean, again, I mean, I think this is something, I mean, so, you know, we have protocols that are open. So there's a young investigator I've worked with very closely named Erin Schenk, who came to the Mayo, who's really interested in this as well. And we have a protocol that's open to actually re-biopsy patients. It was initially written by Bob Doble to look at EGFR, but we've now extended it to ALK, and we've now extended it to patients on immunotherapy. So I think the places, you know, I think that's where, again, Ross is really outstanding in terms of reaching out to patients and being able to get tissues.
Right.
These are always complicated things because there's a lot of demands for these. So clearly at biopsy, a lot of the tissue goes for, you know, particularly these days, goes for genetic and immunoprofiling.
Right.
So I think so we sort of get what's left over, and we have some samples. Obviously, ALK is not, you know, is anywhere between 4 to 7% of all lung cancer patients. So but I think we're, you know, known as a center for that. So I think we're doing the best we can. Right. And I mean, obviously, the more, you know, the tissues we get would be wonderful. And in particular, this ability to re-biopsy tissues and to actually look to see if the complement pathway is actually engaged. This is a challenge to everybody. I mean, I know Trevor Bona has been doing this at UCSF with huge resources and has a paper that is, you know, either in or close to publication, but they only had one ALK patient.
Right. We've got members that don't necessarily go to University of Colorado, you know, see Dr. Camidge or Dr. Doble, but you know, that said, they would still love if they're getting a biopsy, which we've got patients getting biopsies all the time.
I mean, we've actually, I didn't have time to talk about this, but we've actually really developed some pretty sophisticated tools to not just look at these tissues in terms of, you know, is complement activated, but we have now new technologies, and I have a postdoc in my lab who's really learned detailed analysis and machine learning to try to understand these tissues and look at progression on disease. So if we get tissues that are sufficient to do these types of experimental analyses, I think that would be a huge breakthrough. We're actually requesting a supplement from the National Institute of Health to use some of these technologies in our complement project, and that's going to be submitted in September, so we'll have a little more resources to not just say, well, complements are activated, but actually really dig down.
So if we had members in our group that are getting a biopsy for whatever reason, is there any way that they can get their tissue to help your research?
I mean, certainly. I mean, again, I think probably the best person to talk to on that. I mean, so the people that are really engaged in that is Ross and also Dara Eisner at our institution.
Right. Okay.
Kind of the gatekeeper of those, and I think you know, she's terrific. So I would urge them to reach out to them.
All right, we had a couple of questions from members, so I'll just read those out, and if you feel like you're not got a good answer, that's fine. So one question was from a member that is very interested in advocacy for getting, you know, funding for research, and the question was, if the House Defense Appropriations Bill, which includes the full $20 million this coming fiscal year for lung cancer, if it is approved in the final version, what research impact do you think that would have?
So, I mean, I think increased funding for lung cancer will obviously, I mean, it obviously depends where the money goes. I think, some of that will probably be used to help clinical trials. And it's always a situation of opportunities. I think if there's additional funds for lung cancer research, there are clearly, I mean, one example of that is the supplements. I think one of the things that we're already seeing a little bit of, and I don't know if it's because of increased appropriations, is the desire of the NIH to sort of expand what people are doing. And in our project, I think that's, I mean, who knows if we'll get enough, but I think the supplement that we are asking for is something that we really didn't have the ability to do a year or two ago. And if we get some funds to do it and are able to hire some people to do this, I think it'll be a really sort of a deeper insight, not just in, and this will be done largely on patient samples. So let me just emphasize that the supplement is not for more mouse studies. It's for really focusing more on clinical samples. So I do think it'll make an impact.
Terrific. And then we had a question from another member. From your experience, and again, you may not really be in the situation to answer this, do you think if someone has both ALK and EGFR, and they've got a high PD-L1, in this case, they were talking 90%, would immunotherapy like Keytruda be an option?
It's turning out likely that it's not really working very well in either EGFR and in ALK. And I don't think it's really related. So, what determines whether immune response happens? I mean, one issue is how many mutations, how easily these tumors are recognized by the immune system. I think there's a lot of, I don't think we have a very good understanding of why ALK positive patients are resistant or EGFR patients are resistant.
Right.
I think that it doesn't work. The response rates are very low. And there's a lot, this is, not uncommon. I mean, there are now literally, as we've been talking here, there are probably another 20 papers that have been published saying response rates are low, what are we going to do? And I think there's a huge amount of energy looking at this, but I don't think we know. So I would say a patient, even with a high PD-L1 level, who's an EGFR mutant or an ALK positive mutant, I would say the likelihood of success of immunotherapy with checkpoint inhibitors is not great.
Okay. And then a question that I've got, the research that you've been doing, which is kind of out related, would you think that the results can more or less carry over to another oncogene such as ROS1?
Yeah, so one of the reasons that we focus so heavily on ALK, is because we have good models for it. So interestingly enough, there aren't very good models for EGFR. There's some models for EGFR-driven lung cancer. There's very few models for ROS-driven lung cancer in mice. So, you can look at the response of ROS-positive cell lines in human cell lines, but if you're really looking at therapies that target the ability of the immune system to eradicate tumors, you really are forced to do studies in mice, and you're really forced to do studies in immunocompetent mice. So the kinds of experiments where people, and we've done these also, and take human cells and put them into, mice that don't have an immune system, those and test therapies, those are not useful for what the approaches that people who are doing immunotherapy are.
Right, yeah. Very good. Well, you've answered an awful lot of questions and given an awful lot of information. And
I really appreciate your time, and I really, again, want to thank you personally. I think it's, I was amazed, to be honest, at reach out to extend this, and I'm really touched by the level of support, the level of enthusiasm. For me, the ALK positive group, again, I'm a basic scientist. I live in a weird lab. It is always, to me, wonderful to be able to engage the patients, and it's really wonderful to see their enthusiasm, their support, and I want to thank all of, not just for the money. The money is obviously useful, but it really, I think that being able to feel the support is really inspirational.
That's terrific. And then if I understood correctly, you had mentioned earlier that our grant basically allowed you to do work that then let you get more funding for other research.
Complete. So we were, I mean, in particular in this case, the complement, so, you know, truth in advertising, we submitted a complement grant to the NIH, and it was, you know, it was not poorly received, but certainly not funded. And we were getting a little discouraged. We submitted a grant to the Department of Defense, which got very, very close, but did not get funded. And then we got the support from Longevity, and that changed the whole ballgame. So it's really been, you know, it's been fabulous.
That's so exciting for us to hear, because obviously we want your research to lead to, you know, clinical improvements and improved clinical outcomes for our members. But, you know, at the same time, knowing that our funds are actually leveraging other funds is really exciting. So that's great.
No, for us, it was literally made the project. I mean, I would tell you honestly that if we had not gotten support from Longevity, it would have been difficult to continue for much longer.
Terrific. Well, I still haven't met you in person, but I feel like we've met now.
Yeah, close enough. Well, it's been wonderful. I really appreciate your time again, and thank you so much. And it's a pleasure and an honor to work with you guys.
Well, thank you very much. I know that we'll get this put up somewhere with a link, so you'll be able to see yourself on YouTube or Vimeo.
That could be scary. That could be pretty scary.
And our members, I'm sure, will just be absolutely thrilled. So thank you once
again. We're happy to reach out and talk to you. Thank you so much again.
Great, thank you. And we wish you good luck with the rest of your project.
Thank you again. Bye-bye.
Okay. Bye-bye.